Yeah, this is Dr. Alok from Saudi Arabia. So today I'm here to talk about how we can take out genetic material, that is DNA, from the tooth and do analysis of this to help that will help in kindly mute. I can hear some other voices coming. Okay, so basically we are taking out the genetic material from the tooth and how it can be helpful in identifying the individual. So conventionally what we do, we compare the ante-mortem and the post-mortem records in forensic identification of the individual. Now what is ante-mortem record? What is post-mortem? The records what we are making once the person is dead, that is post-mortem records, and the ones which are already available with the dentist, that is the ante-mortem record, that is when the person was alive. So we compare these records, the ante-mortem and the post-mortem records. This is how forensic identification used to be done. Okay, but nowadays a lot of studies are done at molecular level. We have been able to find the DNA structure of the individual, and we have found that there is polymorphism. Each DNA structure is unique for each individual, which can be used for identifying the individual. Now today's presentation, what we are finding out is that whether we can take out the DNA material from the tooth. Is it feasible to take out the DNA material from the tooth? One thing. Secondly, and why we have to take DNA from the tooth? There are so many other places from where we can take. So this presentation is basically about this, the feasibility and the rationale in using this DNA from the tooth for analysis. Now these are the different sources from where the DNA can be taken. The most common ones are the blood, saliva and the semen. But these are the most common ones, but they are less stable sites. That is what it withers away easily over in this site. What more stable site is your tooth or the bone. What is the advantage of tooth and bone over here is that there's in the case of tooth we have enamel and dentine. It acts as a shield or armor, we can say, that protects the DNA material inside so that the DNA does not get affected by the surrounding extreme temperature or climatic conditions in the surrounding. Having said that, let us assume that we have got this mandible with a tooth. Okay, after a long time, say about 10 years, 10 to 15 years, can you extract DNA from this? While the studies have done by TC Bose, he could extract the DNA even after 80 years. Okay, why? Again, because it acts as an armor in which the DNA stays inside without getting destroyed. Even it withers, it does not wither away with extremes of temperature. Suppose there is a temperature of 4 degrees Celsius up to 6 weeks. This study was done by Schwartz, and he found that still the DNA can be extracted from the tooth. So this is the advantage of tooth. Because of the shielding effect, the DNA does not get decomposed or destroyed over there. But before we go for this DNA analysis, I just want to brush up your knowledge about the chemical structure of DNA. I know you during your undergraduate days or your postgraduate days, you must have studied this. Just to brush up your knowledge, what is this DNA made up of? There are two strands in this. Okay. Now these two strands are made up of polynucleotides, or we call it many nucleotides together. What is this nucleotide made up of? It is made up of a sugar, a base, and a phosphate group. Okay. So what are these bases? We broadly classify them as purines and pyrimidines. As a rule, always adenine goes with thymine. That is, this blue one goes with the orange one. If you see here, the blue one bonds with the orange one. And the guanine always bonds with the cytosine. The green one always goes with the red or the maroon one. If you see, green one always goes with this. Now these are how the two strands stand together or stay together because of the covalent bond between AT or CNG. All of us know the structure of the DNA. Now everybody says it's a helical structure, it's a double helical structure. Fine, but when was this DNA structure given or who gave this? Okay, even though the DNA was isolated from the tooth long before, it took around 100 years for the scientists to give this DNA structure. What we easily say nowadays, it's a helical structure. Now, who has given this structure? Everybody's mind, the name Watson and Crick comes. Okay. Okay. So, but Watson and Crick, even they got Nobel Prize for giving this DNA. But very few people know about these unsung heroes. So, who were they? Well, it was a scientific race of all time. It is considered as a scientific race of all time, when all the scientists were trying to find out what is the basic structure of DNA. There were two groups of scientists which were much ahead in time. One was the Watson Crick people, and the other group was of Wilkins and Franklin, who were working on the DNA structure. In this, Franklin's almost got the DNA structure devised. Okay, she was about to give the DNA structure, but his associate or her associate Wilkins leaked the proof or the X-rays to these two people, Watson and Crick, and they were the one who came up with the DNA structure. So sadly, she could not get the due credit, and the whole credit went to Watson and Crick. Okay, leaving this story behind, we'll come back to the history. The DNA, when it was isolated, it was in the year 1869. As I told already, it took around 100 years, around 90 to 100 years for Watson and Crick to give the DNA structure. Okay, it was only in the year 1985 that we found that there is DNA polymorphism. So what do you mean by DNA polymorphism? By this, what we mean, the DNA for each individual is unique. That means my DNA will not match with yours will not match with others. Everyone has his own unique pattern, and it gets transmitted from parents to the offspring in Mendelian pattern. So each DNA is something like a barcode. Your DNA is unique to you only. So what are the potential applications of this? It could be used for finding out the parentage of the child, or it could be used in a crime scene. So what are the ways by which the forensic department uses this? It can be used for DNA profiling. Conventionally, what we are using was the comparison of the antemortem or the postmortem required. Nowadays, we are going for DNA profiling or DNA fingerprinting. Now, first, let us have a look at this. What are these antemortem records, postmortem? How do we compare them? For doing the comparison of antemortem and the postmortem records, three things are required. Now, first of all, that the patient or the victim who is no longer there, he should have gone to the dentist before. That is the first thing. That means that's how you will have an antemortem record. If he had not gone to the dentist before, you won't be having the antemortem record. That is the first thing that is required. Secondly, the more the treatment that has been done on the patient, it is good for us. That means more restorations or fixed prosthesis has done, because it helps in better identification of the individual. Or if there's an anomaly or pathology around. These all helps in finding out, sorting out the individual based on these peculiarities. So first thing, there should be an antemortem or the postmortem record. For that, what should happen? The patient or the victim should have gone to a dentist. Secondly, what should have been there? Multiple restoration or multiple treatments should have been done for him, or some unique pathologies should have been there. It could be a cystic lesion. It could be some, see that peculiar space. What is here is like a sac-like space. That helps in identifying and matching the antemortem and the postmortem records. Another example of antemortem and postmortem record. So what is the peculiarity here? If you see, first of all, the patient or the victim, now he has had treatment done. Okay, so we have antemortem record here. The peculiarity is that if you see here, that's a 3-unit bridge. Again, a 3-unit bridge can be seen here. And the restoration here is something different. It's something peculiar over here. So this helps in identifying, okay, this X-ray belongs to this person. So this is how the victim can be identified. One more case. Now in this case, the lady's face is totally burnt. Okay. Now these are the different postmortem records which have been made. It's not always necessary that antemortem X-rays you will have only periapical. Sometimes you might have only OPG or an orthopontomogram. What you see here, what is special here, you have seen two premolar which have RCT treated. If you look over here, again in the antemortem record, you can see two premolars are RCT treated. In case of lower segment or the mandibular arch, what you see this, some peculiar shape of the restoration is there. Okay, that can be seen over here also. So nowadays we have softwares also. What you do with these softwares? In this software, you put the postmortem record. Take the X-ray, postmortem X-ray, and put there. already it has a bank of all the antemortem records over there. So what it does, it compares with our scans for all the antemortem records with the postmortem record, and any similarity it finds out, it comes out with a result. So it has become relatively easier with this software in identifying the individual by comparing the antemortem and the postmortem records. Now, having said that, if you can compare the antemortem and the postmortem records and identify the individual, then what is the need for DNA fingerprinting? And that too from the tooth. Why do we need all this? Because of there are some disadvantages of this comparison. What are these disadvantages? Let us have a look at them. The first one is this can be a discrepancy. What do you mean by this discrepancy? Means suppose a patient comes to a dentist, there's a deep caries lesion, he advises for endodontic treatment, and he has taken an X-ray. Now he has a pre-X-ray over here. Without doing any endo treatment, the patient doesn't come back. He goes to some other place where the treatment has been done, and that fellow does not take X-ray that time. So in the post-mortem X-ray, there will be an RCT treated tooth, but the X-ray what you have, it won't have a treatment done. So this is what we mean by discrepancy. The antemortem and the post-mortem X-ray will not match. Poor quality of X-ray. What do you mean by that poor quality? Suppose while taking the X-ray, there was too much developer solution or less fixing solution or some cone cut is there, and you cannot appreciate the quality image of the X-ray. This is the problem. Or you cannot inability to locate the person. Suppose the patient is from Dubai. Now he has gone to Sharjah or UAE for visit. So some mishap happens in Sharjah or UAE. So Dubai people cannot locate the records, or the Sharjah people cannot locate the records because he has gone to some other city. In the worst situation, suppose someone has come for tourism to Dubai from some other country, say India or UK, and now what happens? They will definitely not be able to find out the X-rays. Since these are all the voids which can happen while using the comparison method. So how to overcome all these voids? Go for DNA profiling or DNA fingerprinting. To understand how this DNA analysis is to be done, we need to know some basics. I know at your undergraduate level you might have read all this. Definitely you should have read. Just to brush up your knowledge, if your people are around, what is chromosome? What do you mean by chromosome? Now, chromosome basically is an organized package of DNA in the cell. You should know there are two types of cells. You have the prokaryotic cells and the eukaryotic cells. In case of eukaryotic cells, we have the nucleus. In that, we have the organized package of DNA. Now, what is gene? Now, if you see this chromosome, where it is in the cell, in the nucleus. Now, this chromosome is made up of long strands of DNA. Now, gene is just a segment of this DNA which helps in coding for proteins. Now, this is called as the coding DNA. This forms only 2 to 5% of the total DNA. The function of the remaining 95% of the DNA is nothing. Now, this is known as the non-coding DNA or the junk DNA. Okay, now I already have told what you mean by junk DNA. This is the non-coding DNA, which has no work. Now, what are these tandem repeats? Now, if you have a look at here, what you see, this is DNA. DNA, in this we have a gene. It's a segment of a DNA which helps in coding for proteins. Now, this forms only 2 to 5%. Remaining 95% has no use. It has no role. This is called as a non-coding DNA or the junk DNA. This importance of this is that it keeps on repeating, and the pattern of repetition of this non-coding DNA is unique for each individual. So basically, now what are tandem repeats and how these tandem repeats are unique? I told you we have two types of DNAs. We have the coding DNAs and the non-coding DNA, which is also called as the junk DNA. Now it is the junk DNA which keeps on repeating. This is called as the tandem repeats. Can be at mini satellites or short tandem repeats or long tandem repeats. Now these short tandem repeats are unique for each individual. So the repetitive DNA sequences in the non-coding DNA, these are called as the tandem repeats. It can have satellites, mini satellites or microsatellites. It is this mini satellites which is unique for each individual. So till now what we have learned, a cell can have nucleus or cannot have nucleus. If it has a nucleus, it's called a eukaryotic cell. In this we have the chromosome. What is chromosome? It is an organized and a condensed package of DNA. DNA appears as long strands. Okay, a segment of that is gene which codes for protein, that is the coding DNA. Rest of the DNA which does not code for DNA is called as junk DNA. Now it is the junk DNA which has the tandem repeats. The mini satellites is important for us. We have to look for these mini satellites. The pattern of repetition of this mini satellite is unique for each individual. So it becomes your ID card. So what are the oral sources from where DNA can be retrieved? It can be retrieved by teeth, by saliva, or by biopsy. How do you retrieve from teeth? You can go for the easiest method is go for an endodontic access to remove the pulp from which the DNA can be extracted. From saliva, the easiest way would be to use a micropipette or use a filter paper. From biopsy, how do you retrieve the material? By incisional biopsy, excisional biopsy, or a aspiration biopsy. Now, from teeth, what type of cells you are getting? The most important one is the pulp, where all type of cells you get. From the saliva, what you get? You get the desquamated epithelial cells, and from the biopsy material, you get the epithelial and the connective tissue cells. Now let us have a look at the tooth. Is it a feasible material for retrieving DNA or the cells? For DNA, what you need? You need any cell which has a nucleus. So do we get nucleated cells in the teeth? Well, if you ever look at the enamel, it does not have any nucleated cell. If you look at the dentine, do you think it has any nucleated cell? But in case of dentine, we have odontoblastic processes. In these odontoblastic processes, we can have nucleated cells. So that can be used. But the major portion, what is here used is this pulp. In this pulp, we have the different types of cells. We have the fibroblasts, odontoblasts, epithelial cells, undifferentiated mesenchymal cells, and peripheral neural cells. A lot, and in addition to this, even the blood is there. So definitely you can get the nucleated components of cells from here, that is from the pulp. In addition to this, we have some less stable sites. What are those? We can have the periodontal ligament fibers, we can have the adherent bone, or there can be cementum. If you remember, there are two types of cementum, cellular cementum and the acellular cementum. It's the cellular cementum which can be used as a source of DNA. The most stable site would be definitely be this pulp, which is enclosed by enamel dentine from outside. Now have a look at this. This person was buried for since long time. Okay. Now from this tooth, can you have sufficient amount of DNA for analysis? Well, it depends on a lot of factors. The most important is the size of the pulp chamber. If you look at the anterior, the size of the pulp chamber is small as compared to the posterior teeth. So definitely the posterior will have larger pulp size, so sufficient amount of DNA material will be there. This is one thing, but this holds good for a freshly extracted teeth, that you get sufficient amount of DNA material. What about such cases? First of all, what is the amount of DNA required for an analysis? We require only 1 nanogram for doing DNA analysis. And how much we get from here? It is around 10 to 20 milligram. Mind it is milligram. What is required? It is nanogram. So normally we get sufficient material from tooth for analysis. Sometimes if the material, if the body has been there buried for long time, okay, and you are not able to get 1 nanogram material, then you can go for PCR method to amplify the amount of DNA what you have. Now there are different factors from which makes which are the responsive for the amount of DNA that can be extracted. First of all is the type of tooth. Now in sizes, the pulp chamber size is smaller, so you get less DNA. In case of molar, the pulp chamber size is bigger, so you get more DNA or the cellular elements from here. The degree of decay, if the tooth is totally decayed, the amount of DNA that can be removed will be negligible compared to a healthy tooth. Definitely you get more DNA from here. So if trauma is there, if the teeth has a trauma, there is an open pulp, you won't be able to retrieve much because the tooth would have become non-vital and the pulp fragments will not be there over there. Again, the period of time from extraction of DNA, from extraction to DNA isolation. A freshly extracted teeth definitely will have more cellular elements compared to one which has been there buried for long time. What about the human cycle? What is the changes in the teeth? What you see? These are basically the aging changes. In case of young, the pulp chamber is bigger. As the child becomes adult and then it becomes old. In old, the pulp chamber becomes very small. They'll be stenosed, calcified. They won't be able to get enough DNA material from that. So younger or adult people, you get better chance of getting analysis because you have more amount of cellular elements over there. Now, coming to the steps involved in DNA. First, we talk in general how we do from a blood. From the DNA from the blood, first we have to extract the DNA. Now, DNA is a long fragment. It has to be broken down into small fragments. This is done by DNA digestion. Now, these fragments are then transferred onto a agarose gel. This is done by electrophoresis. Then from here, from the agarose gel, it is transferred to a nylon membrane. Why? Because this is fragile and this nylon membrane is more stable side. So we get the negative side and the larger ones go towards the positive side. Now, as I have told this, now where this electrophoresis is done, it's done on an agarose gel. Now, agarose gel is not stable. It is very fragile. So this has to be transferred to a stable medium. What is used here is a nylon membrane, which is done by a method called as southern blotting. The DNA is fixed to this with heat to the nylon membrane. So far what we have done, we have taken a blood sample or we have taken pulp. From the pulp, we have taken out the DNA, done electrophoresis where the fragments are divided from small to big ones. This is not stable side. So we take it to a nylon membrane from the agarose gel, which is more stable. That is done by southern blotting by fixing the membrane with UV heat. Yeah, okay, ma'am. So now we have here. Next, we have to add the label probes over here. That label probes gets attached to it and we can visualize them. Now the DNA probes are attached to this. You have to put X-ray. With the X-ray, you can visualize this as hybridization technique. These are different bands which can be visualized. Now, what is PCR? This is basically how the DNA has been analyzed. Now, sometimes if you don't get 1 nanograms of DNA, then in such cases, you can go for PCR technique. Basically, what does this PCR do? It is like a photocopy machine, whereby running through multiple cycles, you get sufficient amount of DNA. So from one strand, if you see here, if you run for multiple cycles, you get multiple long strands. Multiple cycles will lead to millions of copies which can be used for PCR analysis. This is how the DNA analysis is done. There are different steps which get taken. What is the application of this? It can be used where DNA is not available from any other part of the body, like in case of plane crash, charred bodies, or building collapse. In all such cases, you can go for DNA fingerprinting because DNA from the most stable sites is required. Here, the tooth acts as a shield, so it easily can be saved over there. Other less stable sites like blood, it will not be available there. So we dentists are no less like a James Bond, which helps in identifying the person. So to summarize how we are doing, first from the DNA is extracted, broken down in small fragments, put on the electrophoresis, transferred to a more stable membrane, then X-ray is done to get the auto read, where all the probes can be used to see the tandem repeats. To conclude, if you see this picture, what I want to say here is that this sunrise shows the future of the use of DNA fingerprinting. We have a bright future, but this path towards this tide shows the obstacles. Now, what are the obstacles we have here? We don't have sufficient equipments. We need to work on that. The expertise who are to work on this, the knowledge among the forensic people, all this has to be taken care, and definitely in the future, DNA fingerprinting from the tooth will be done. Meanwhile, the golden standard still remains the use of the comparison of antemortem and the postmortem records. Thank you. These are my references. Thanks.